So there's obviously a whole, now this was not really a starting point. We also started earlier with other, molecular levels. We looked at microRNAs also in one project. We looked particularly also at neurofilament light chain, because that is kind of a hot topic in the neurological diseases. But we currently try to take, an outer perspective on what is basically circulating in the blood, how can we detect it in a broader scale, and what kind of questions can help us answer that? What is the unmet clinical need that we have in daily clinical practice? And how can we help our patients best? Where do we fit in? Because there's neuroimaging and there's also other techniques, obviously, but still we don't have any stroke context-specific blood-based biomarker compared to many other diseases, obviously, such as a myocardial infarction, where we would do troponins, such as prostate cancer, where we do PSA. These biomarkers are around for ages, but for stroke, we don't have anything. And it's just very unlikely that there is nothing. There's thousands of metabolites, proteins circulating, and in our daily clinical routine assays, we just test for 20 of them or so. So there's really a large window of opportunity there. And we just currently have the technologies that they just developed over the last couple of years, as metabolomics, proteomics, and so on, different ways of performing proteomics. So we believe that there's a strong window of opportunity now compared to also earlier times, when people started looking at this, because obviously that's not what we came up with a question for now, developing a blood-based biomarker for stroke. So we have established a large cohort of patients, overall accumulating 3,000 patients with suspected stroke, so either ischemic stroke, stroke mimics, hemorrhagic strokes, patients with transient ischemic attacks, and that we followed over time, not only upon hospital admission, but day 2, day 3, day 7 also. And there's, for example, the question that we would very much like to follow early neuronal injury over time. So it's highly unlikely that a patient always from the stroke unit or the ICU even always is transferred back to the CT to see, is he really doing worse in his brain or not? And what we would highly value is obviously a marker that would specifically indicate, is there continuous neuronal injury now? Is it stable? Is it decreasing? We would like to identify individual trajectories of neuronal injury to see also obviously then over time whether there's still a treatment window. We would like to dive into where strokes come from. So stroke etiology is a large topic right now because we still have an issue of identifying the individual cause of stroke in many patients. And this has also therapeutic consequences when it comes to secondary prevention of stroke. So there's many, many questions where I feel that we are not performing very well on a diagnostic level, and that would help us tremendously on treating our patients better. And using our cohort, we would like to profile plasma and serum samples with regards to metabolome, proteome, and other molecular layers, and hopefully come up with the solution for the future.